Hello students, my name is wasi anjum. I teach NEET aspirants zoology. I have done M.Pharm in pharmacology from Jamia Hamdard, and after that I worked as assistant professor pharmacology in Saudi Arabia Pharmacy College, and I also worked here as a guest faculty in Jamia Millia Islamia. There my work also was of a senior research fellow, and my research topic there was heart failure. I also worked as a pharmacovigilance officer in Wipro, and for last 8 years I have been working as a NEET zoology faculty. Today I have to teach you about the blood circulation in humans. So blood is meant for transportation of certain molecules like carbon dioxide, oxygen, nitrogenous gas and nutrients. So for that, blood circulation should take place, that is the repeated circulation, repeated cycles of heart activity, contractile activity. So this we can understand by understanding first the anatomy of heart. That is the structural detail of heart. So the heart is four-chambered, two atrium at the receiving end and two at the exit end of the heart. That is called, the first it is called atrium, the receiving end, and two ventricles at the exit point. This is my right hand, and this is the right side of the left side of the board, opposite left side of the board, and this is the thereby right ventricle and right atrium. So this is right atrium, this is left atrium, this is left ventricle, this is right ventricle. These two valves at the junction of atrium and ventricle are atrioventricular valve, of which the first left side is called bicuspid valve and this is called tricuspid valve. And here bicuspid valve is also specifically called mitral valve, and this is at the end where the aorta starts, arteries starts, so it is aortic valve or semilunar valve. Here also it is semilunar valve at the exit point, so this is semilunar valve. These two are the semilunar valves. Now, here the outgrowth of muscle or ventricle is papillary muscle. Papillary muscle, and the chorditendini joins the atrioventricular valve with papillary muscle, and this chorditendini is of collagen. So chorditendini is collagen containing, that is gives the because of its tensile strength, gives the attachment point or positioning of the atrioventricular valve and preventing the backflow or inversion of the atrioventricular valve. So bicuspid and tricuspid valves are prevented from inversion through these attachments of chorditendini connective tissue. Now here it is SA node, and here it is AV node. So AV node and SA node are the special musculature, nodal musculature, causing the constant firing at the constant rate, the firing of electrical impulses to first atria and then to ventricle down the path, leading to the atrial and ventricular systole or contraction. So these are the things which we study under the anatomy of heart, and after that we start studying about the cardiac cycle, that is the physiology of heart. So in physiology of heart, we will study sequential events of systole and diastole, that means the contraction of heart and relaxation of heart. So these sequential events we will study in cardiac cycle. So cardiac cycle starts with the generation and propagation of action potential in the atrium first, leading to atrial systole. And because the AV valve is spacier and a little bit less in the firing or rate of action potential, so it is limiting, and this delay in the ventricular systole will lead to this delayed ventricular systole due to pacemaker AV node. This delayed ventricular systole will lead to simultaneous atrial diastole. Simultaneous atrial diastole. That means when there is a delay in ventricular systole, this delay the atrium will utilize in atrial diastole. So again, ventricular diastole and ventricular diastole and this joint diastole, both are in diastole. So this is the diastole, joint diastole, the last step of this first cardiac cycle. Starting of the second action potential further takes place, leading to the continuous waves, continuous action potential, and continuous cardiac cycle. So the heart is myogenic. The heart is myogenic in characteristics in human heart, and it is neurogenic, that is regulated with neuron in the case of arthropods. But here it is myogenic under the control of nodal musculature, SA node and AP node, etc. So SA node and AP node, after that joint, after that the bundle phase and left bundle, right bundle, lastly Purkinje fiber, leading to the muscular contraction and cardiomyocyte contraction. And this concerted, well concerted effort of all the cardiomyocytes, that's the contraction and systole of the heart, after that diastole of the heart. So these sequences we have studied. Now I will try to make you aware of the two terms, that is heart rate and cardiac output. So heart rate is expressed as number of beats or number of cycles per minute, and cardiac output is ml of blood pumped per minute, and this ml of blood pumped by each ventricle per minute is called actually cardiac output. So this another one terminology is stroke volume, that is the volume of blood pumped per cycle. So the formula of cardiac output is stroke volume multiplied by heart rate, that is the stroke volume is ml per cycle and heart rate is cycle per minute. So this cycle cancels and leads to ml per minute. So this is the unit of cardiac output, amount of blood pumped per minute. Now talking about the calculation in terms of its values, stroke volume is 70 ml per minute and heart rate is 72 beats per minute. So multiplied together it forms 5000 ml of blood per minute or 5 liter of blood per minute. So this is the cardiac output of normal heart. The athletic heart has somewhat more value of cardiac output. So these two terminologies are understood, and now we will understand about the simultaneous occurring of all these events in one graph, simultaneous presentation of these all activities in one graph, and that graph consists of ventricular pressure first, and then a little bit more of the atrial pressure, so this is ventricular pressure and this is atrial pressure, and one more pressure is there, that is aortic pressure. So this is aortic pressure. In terms of its values, these two ventricular and atrial pressure is near to zero value, 0 mm of Hg, and after that 20, then 40, then 60, and then at the base of the aortic pressure it is 80 mm of Hg, after that 100, and at the peak of ventricular pressure it is 120 mm of Hg. So these values, two values 80 and 120 mm of Hg are the important values of aortic and ventricular pressure. It is also important, this near to zero value, and these three combinations will lead to the opening and closing of the valves, leading to the synchronous pumping of the synchronized contraction and relaxation, leading to the effective pumping of the blood in a cardiac cycle. So we start here with this junction of atrium, ventricle and aorta. Ventricle, atrium and aorta. So two valves here are mitral valves, and two valves, one valve here is mitral valve and one valve here is aortic valve. Aortic valve and mitral valve. So here at this junction of atrium and ventricle, it is not none but mitral valve. So mitral valve also at this junction of ventricle and atrium, so it is also mitral valve, and in the upper region here at this junction it is aortic valve, and here also it is aortic valve. So now talking about the closure of this aortic and mitral valve and opening of this aortic and mitral valve. So here, when the ventricular pressure rises and the ventricle is in contraction, it will lead to forceful closing of mitral valve to prevent the backflow. So it is closure of mitral valve closed. Now here the ventricular pressure is more, so ventricular pressure is more, ventricle is in contraction, leading to the this mitral valve is closed here at this junction, and every aortic valve is forced open when ventricle is in contraction. Forced open aortic valve, so it is open here, and aortic valve is at this junction when the ventricular pressure is decreasing. Here the ventricular pressure was increasing, ventricle was in systole, and here it is ventricular pressure is decreasing or ventricle is in diastole. So in diastole, this aortic valve will lead to closure because of the preventing of the backflow from aorta, and forced closing of the this aortic valve because the pressure here is more and than the ventricle, and forced closing of the aortic valve. So it is closed here. Now here the mitral valve, when the ventricular diastole, when the ventricle is in diastole, relaxed state, so the filling of blood will take place, leading to the forced opening of the mitral valve. So these four phenomena together, and this line here at this first junction, this line at the second junction, this line at the third junction, and this line at the fourth junction, will lead to two strips, formation of two strips here, small strips. The first being isovolumetric contraction, where